Hello, Professor. This is Alfredo Barrera, and today I will be doing the alternative assessment for lecture exam 3. So I want to start off by saying the action potential is essentially a signal, electrical signal that is sent from the brain into the brain or the spinal cord that travels down a motor neuron to the axon terminal, which is essentially the end of the neuron. The axon terminal meets the muscle at a specific area called the neuromuscular junction, which is where the nerve and the muscle connect. When the signal arrives, calcium channels open, letting calcium enter the axon terminal. This essentially causes ACH, which is a chemical messenger, to be released into the synaptic cleft, which is a small gap between the nerve and the muscle. ACH, it moves down the synaptic cleft, shown here. As soon as it starts moving down across the synaptic cleft, it binds to acetylcholine receptors, which are essentially special receptors on the motor end plate, which is the part of the muscle cell that receives the message. These receptors, they sit on the sarcolemma, which is the muscle cell membrane. ACH binds to its receptors. It releases sodium ions, which essentially they go into the muscle and they change the charge of the sarcolemma. That is called also depolarization. So that change, it creates a new action potential that spreads along the sarcolemma, and it moves into the muscle through a T-tubule, which essentially helps carry the signal inside the cell. The signal then reaches the terminal cristae of the sacroplasmic reticulum, which is essentially the storage area for calcium inside the muscle. That causes more calcium to be released into the muscle cell. So calcium, it binds to troponin, which is a protein that sits on tropomyosin, which normally blocks the binding sites on actin. Shown here. When calcium binds to tropomyosin, it moves away, and in myosin, the thick protein filament, it attaches to actin, and this makes the muscle contract. When the signal stops, ACHE, it breaks down ACH in the synaptic cleft. It's just ending the signal. Then calcium is pumped back into the sacroplasmic reticulum. Tropomyosin moves back to block actin, and the muscle relaxes.